40 years from now, could we actually be looking at a world where humans live forever?
Hmm.
It's this huge question, isn't it? Woven through history, myths, and now, well, serious science.
It really is.
So for this deep dive, we've gathered a lot of research, and we want to explore just how feasible this idea is, specifically for you listening.
It's definitely an exciting prospect, no doubt.
Yeah.
And the science is moving incredibly fast. But maybe before we get too far ahead of ourselves, we should probably pin down what living forever actually means, in a scientific way.
Good point.
Because the material we've looked at, it brings up several pretty distinct concepts.
Okay, yeah, let's unpack those. So first up, our sources talk about biological immortality. Now, this isn't about being indestructible like a superhero.
Not at all, no. It's specifically the absence of aging. So imagine your risk of death just doesn't increase as you get older. Your mortality rate basically flatlines age-wise.
Right, it stays constant.
Exactly. But, and this is key, it absolutely doesn't mean you're invulnerable. You could still get hit by a bus or catch a fatal disease. It's just that age itself wouldn't make dying more likely.
Okay, got it. And it's fascinating, isn't it, that nature seems to have sort of cracked this already in some organisms.
Well, yeah, in a way.
Like the Turritopsis dohrnii jellyfish, the one that can, revert back to his younger self.
That's the one. It can revert to the polyp stage. It's an incredible biological reset.
Yeah.
And then you've got freshwater hydras, too.
Yeah.
They seem to regenerate almost indefinitely.
So these are like nature's proof of concept, showing it's biologically possible.
They're definitely intriguing examples, yeah. They show us what can be possible in biology. But the mechanisms are really specific to them. Very complex.
Right.
The cellular stuff in jellyfish, the stem cell dynamics in hydras. We can't just copy-paste that into humans. Our biology is just vastly more complicated.
Okay, so while they offer a glimpse, human solutions probably need a different path. So then what about negligible senescence? That sounds similar.
It's very close, yeah. A close cousin, you could say. Again, the core idea is that your mortality risk doesn't climb with age. You kind of sidestep that wall of mortality that hits most species, including us.
Avoid the wall. Okay, and the third idea.
That's radical life extension. This is maybe less about stopping aging completely and more about, well, significantly stretching out the maximum lifespan.
So living for centuries, maybe. Way beyond what we see now.
Exactly. And you have groups like the SENS Research Foundation. They're aiming for what they call engineered negligible senescence. Their approach is all about repairing the damage, the cellular and molecular junk that builds up as we age.
Okay, so it seems like the real focus for you, based on the research, isn't making us invincible. It's more about tackling aging itself, aiming for that negligible senescence or a really major extension of healthy life.
That's the main thrust, absolutely.
Now, that 40-year time frame we mentioned, it feels, I don't know, both really long and also maybe not long enough for something this huge.
It definitely creates a tension, doesn't it?
Yeah.
You've got this incredible acceleration in tech, AI, biotech. Things are moving so fast.
Right.
But then you have the sheer complexity of human biology. And honestly, the time it takes to get something from a lab discovery to an actual safe treatment for people, that's often a very long road. Slow.
Yeah, the translation gap. We've seen huge shifts before, though, haven't we? Antibiotics, vaccines. They totally changed the game for human health.
Absolutely transformative.
So the question is, are we maybe on the brink of another shift like that? One that could overcome what one source called the evolutionary basis of limited longevity. And could that happen in the next 40 years?
That really is the crux of it, isn't it? Are we at that kind of inflection point? And to even start answering that, we need to dig into the science of why we age, what's actually going on.
Okay.
Because the sources are clear. Senescence, aging, it's not just one thing. It's this incredibly complex web of changes happening right down to the molecular and cellular level.
Right. Let's get into those mechanisms then. The research keeps bringing up these hallmarks of aging. What exactly are those? Why are they so central?
So think of the hallmarks as maybe the main culprits, the key biological processes that start to go wrong over time. They're what drives the decline we associate with getting older.
Okay.
Initially, scientists identified 9 of them, but, science moves on, we learn more, and now the list is often expanded to 12. It shows how our understanding is deepening.
12 interconnected problems, basically.
Exactly, interconnected. That's the crucial part. They don't happen in isolation. They all influence each other, like a tangled web.
Okay, a web, not just separate issues. So let's look at a few. Genomic instability, that sounds bad.
It represents a constant battle our cells face every day. Just through normal living, metabolic processes, environmental factors, our DNA takes a beating.
How much of a beating?
The estimates are pretty staggering, potentially up to 1 million damaging events per cell per day.
Wow, 1 million per cell.
Yeah. Now, our cells have repair crews, repair mechanisms, but they're not perfect. They miss things or they make mistakes. And over a lifetime, this damage accumulates.
And that leads to?
Mutations, obviously, which increases cancer risk.
Yeah.
But also just general cellular dysfunction. Cells don't work as well, or they might enter that senescent state we'll talk more about.
Yeah.
So, yeah, this constant damage is a fundamental drag.
Okay, 1 million hits a day is hard to wrap your head around. Then there's telomere attrition. I picture these like the little plastic ends on shoelaces, right? Protecting the chromosome.
That's a perfect analogy. They're protective caps. The issue is, due to how DNA gets copied when cells divide, these telomeres naturally shorten a bit each time.
Like a countdown timer on the cell?
Sort of, yeah.
Yeah.
When they get too short, critically short, the cell faces a choice. It can stop dividing permanently, that's called replicative senescence, or it can trigger self-destruct apoptosis.
But isn't there an enzyme, telomerase, that can fix them?
There is. Telomerase can rebuild telomeres. But here's the catch. In most of our adult body cells, its activity is deliberately kept very low, suppressed.
Why?
It's thought to be a trade-off, an anti-cancer mechanism, because cancer cells often reactivate telomerase, and that helps them divide uncontrollably. So suppressing it protects us from cancer early on, but the shortening contributes to aging later.
Wow, a real biological dilemma. Protect from cancer now, contribute to aging later. And I saw something fascinating, too, that chronic stress, like psychological stress, seems linked to shorter telomeres.
Yes, that connection is being actively researched. It really highlights how our mental state can influence these fundamental biological processes. It's not just physical wear and tear.
Mind-body connection right there. Okay, moving on. Epigenetic alterations. This one feels a bit more abstract than DNA damage or telomeres.
It's a bit more conceptual, yeah. Think of your DNA sequence as the hardware, the basic instructions. The epigenome is like the software tells the hardware which instructions to run, when, and how strongly, without actually changing the DNA code itself.
So it's about gene regulation.
Exactly. It involves chemical tags like DNA methylation or modifications to proteins called histones that package the DNA. These tags can be added or removed, and they're dynamic. They respond to our environment, our lifestyle.
Okay. And how does this relate to aging?
Well, as we age, the patterns of these epigenetic tags tend to drift. Some genes might get inappropriately silenced, others inappropriately activated. You see changes in methylation patterns, for instance. This messes up the normal gene expression needed for healthy function.
And can we track this drift?
Yes, that's one of the really exciting areas. Scientists have developed epigenetic clocks based on these patterns. They can measure your biological age, which might be different from your chronological age.
And the really big hope here
is reversibility. Because these tags aren't permanent changes to the DNA sequence, there's a potential, theoretically at least, to reset them, to nudge them back towards a more youthful pattern. That's a major therapeutic target.
Resetting the software, that's powerful. Okay, then cellular senescence. We mentioned the zombie cells.
Yes, the zombie cells. That's a catchy term. So these are cells that have stopped dividing, usually due to damage or stress, but they resist dying off. They just hang around.
And they're not just passive bystanders.
Far from it. They actively secrete a nasty cocktail of inflammatory molecules, enzymes that break down tissue, growth factors. It's called the SASP senescence-associated secretory phenotype.
Sounds bad.
It can be, especially when these cells accumulate. This SASP contributes to chronic low-grade inflammation, sometimes called inflammaging damages nearby tissues, and is implicated in a whole host of age-related diseases, from arthritis to neurodegeneration.
But I read they sometimes have a good side.
They do, yes. In the short term, senescence can be beneficial. It helps stop damaged cells from becoming cancerous, plays roles in wound healing, even embryonic development. The problem is the chronic accumulation with age. What was protective becomes detrimental.
Right. Useful short-term, harmful long-term. Okay, what about stem cell exhaustion? That sounds pretty fundamental, too.
Absolutely fundamental. Stem cells are our body's internal repair kit, right? They can divide to make more stem cells or differentiate into specialized cells to replace damaged ones, essential for tissue maintenance.
But they run down over time.
Yeah, essentially. As we age, the number of stem cells in many tissues decreases, and the ones that remain often don't function as well. They become less efficient at repairing damage.
So our ability to regenerate just declines.
That's a big part of it. This exhaustion impairs tissue repair, contributes to organ dysfunction, and is a major factor in age-related frailty.
Makes sense. If the repair crew gets smaller and tired, the building starts to crumble. And the last one in this initial group, mitochondrial dysfunction.
The cell's power plants, right?
Exactly, the powerhouses. Mitochondria generate most of the cell's energy currency, ATP, but with age, they tend to become less efficient.
Meaning less energy. Less energy production, yes. But also, they start producing more harmful byproducts, like reactive oxygen species, free radicals. This increases oxidative stress, damaging the cell. Plus, mitochondria are involved in lots of signaling pathways, so when they malfunction, it throws off other cellular processes too.
Okay, wow. It's really clear hearing you describe these. It's not just one thing going wrong. It's this whole cascade, this network you mentioned.
That's absolutely the key takeaway. It's a systems-level problem. All these hallmarks are interconnected. They influence each other.
So you can't just fix one and expect to solve aging.
Probably not entirely, no. You might get some benefit, but it's unlikely to be a magic bullet. For example, you could use senolytics to clear out senescent cells, which sounds great.
Yeah.
But if you still have high levels of genomic instability creating new damage, or your mitochondria are still sputtering, then new senescent cells might just form again, or other problems will persist. It strongly suggests we'll need therapies that target multiple hallmarks, probably in combination.
A multi-pronged attack. Still, the big advance here seems to be identifying these hallmarks. It gives science actual targets to aim for, doesn't it?
Precisely. It's shifted the field. Instead of just describing aging, researchers now have concrete biological processes they can try to intervene in. It provides a framework, a roadmap, for developing anti-aging strategies. It's a huge step forward.
But, okay, even with this roadmap, our research stresses that aging isn't, one size fits all. We all age differently. What drives that variation?
You're absolutely right. It's highly individual. And it comes down to this complex interplay between, well, three main things: our genes, our environment, and just random chance events accumulating over a lifetime.
How much is down to genes versus everything else?
The estimates vary a bit, but generally, studies suggest genetics account for maybe 20 to 30% of the variation in lifespan.
Only 20, 30%.
Yeah, which means the lion's share, 70 to 80%, is down to non-genetic factors. And a huge chunk of that is lifestyle and environment. Diet, exercise, stress, exposure to toxins, sleep. All these things play a massive role.
That's actually really empowering, isn't it? Knowing that so much is potentially influenced by our choices. We saw that study from 2023 on U.S. veterans.
Oh, yeah, that was striking.
It suggested that adopting 8 specific healthy habits could potentially add something like 24 years to life expectancy. That's huge.
It is remarkable, and it really highlights that powerful strategies are already available to many of us right now. It also hints that personalized approaches will likely be key in the future, tailoring interventions to an individual's genetics, lifestyle, and specific aging patterns.
And there was another concept mentioned, resilience. What does that mean in the aging context?
Think of resilience as your body's built-in ability to maintain itself and bounce back from stress or damage. It's all those complex maintenance and repair systems working behind the scenes.
Okay.
The idea is that aging occurs when the cumulative burden of damage starts to overwhelm these resilience mechanisms. They can't keep up anymore. So this suggests that maybe effective anti-aging strategies need to do more than just repair damage.
They need to boost the systems themselves.
Exactly. Bolster that innate resilience, enhance the body's own ability to cope with and repair damage as it occurs. It's about strengthening the foundations, not just patching the cracks.
Strengthening the foundations. I like that. Okay, let's shift gears then to the really cutting-edge stuff, the frontiers of longevity research. What are some of the most exciting avenues being explored right now?
Well, one area that's generated a huge amount of buzz is targeting cellular senescence with drugs called senolytics.
Right, hitting those zombie cells we talked about.
Exactly. The rationale is simple. These cells accumulate with age. They secrete harmful stuff, so let's get rid of them. Senolytics are designed to selectively trigger apoptosis or self-destruction in these senescent cells while sparing healthy ones.
And we mentioned that early combination, dasatinib and quercetin, D and Q.
Yes, D and Q was really pioneering in this space, and the results in animal models, particularly mice, have been frankly quite remarkable.
How remarkable.
Well, some studies showed lifespan extension in older mice by as much as 36%. Significant improvements in various age-related conditions, too. Really dramatic effects.
36%. Okay, that's massive.
Huh.
But the big question, how has that translated to humans? Are we seeing anything similar?
Well, that's where it gets more complex. The human trial results have been, let's say, more nuanced so far.
Meaning?
We've seen some encouraging signs in specific contexts. For example, some studies in patients with idiopathic pulmonary fibrosis, a lung disease linked to senescence, showed improvements in physical function. Another study showed senolytics could clear senescent cells in people with diabetic kidney disease.
Okay, so some positive signals.
Yes, but other trials have shown more modest or even transient effects. A study looking at bone health in older women, for instance, found only subtle benefits that didn't last.
So that translational gap we mentioned earlier is definitely visible here. Why the difference between mice and men, so to speak?
It's likely a combination of factors. There are fundamental biological differences between species, obviously. Human aging is vastly more complex than mouse aging. Plus, human trials are often done in older individuals who already have multiple health problems, which complicates things. Mice in labs live in very controlled environments.
Right, much simpler system.
But despite these challenges, the development of senolytics is still seen as a major step. It's a paradigm shift because it's one of the first approaches trying to target a root cause of aging, not just manage symptoms of diseases.
Targeting the cause, not the symptom.
Exactly. And the NIH is investing heavily in this with initiatives like Senate, aiming to map out where and when senescent cells appear in the human body. Oh, and there's also an alternative approach called xenomorphics.
Xenomorphics.
Yeah, instead of killing the senescent cells, these drugs aim to change their behavior specifically to stop them from secreting that harmful SESP cocktail. Maybe a gentler approach.
Interesting. Neutralizing them instead of eliminating them. Okay, what's another major frontier? Epigenetic reprogramming sounded almost like science fiction earlier.
It really does feel that way sometimes. So we talked about epigenetic alterations being a hallmark, but also potentially reversible. Some researchers are quite bullish, suggesting epigenetics might be the main determinant of lifespan.
And this ties into the Yamanaka factors.
Precisely. Shinya Yamanaka's Nobel Prize-winning discovery showed that just 4 specific factors could turn adult cells back into pluripotent stem cells, iPSCs, essentially wiping the slate clean, epigenetically speaking.
Incredible. But didn't we also say doing that in a whole living organism is risky? Cancer risk.
Absolutely. Full reprogramming in vivo is generally considered too dangerous right now because of the risk of uncontrolled cell growth, tumors. So the big focus currently is on partial reprogramming.
Partial? How does that work?
The idea is to apply the Yamanaka factors, or similar reprogramming agents, only transiently for a short period. The hope is that this is enough to reset some of the age-related epigenetic marks, rejuvenate the cells, restore more youthful function, without pushing them all the way back to a pluripotent state and risking cancer.
A controlled reset, not a full wipe.
Exactly. It's an area attracting massive investment. Companies like Altos Labs, Jeff Bezos' venture, and Retro Biosciences, backed by Sam Altman, are pouring huge sums into this.
Big names, big money.
Yeah, which tells you something about the perceived potential. But the challenge is immense, figuring out how to do this safely, effectively, controllably, throughout the tissues of a complex organism. It's vastly harder than doing it in a petri dish. And we shouldn't forget lifestyle, diet, exercise. They also impact the epigenome, so it might be a combination approach again.
Huge potential, but major safety and delivery hurdles still.
Got it. What about directly intervening in our genes? Genetic interventions like gene editing or telomere stuff? Right. So gene editing, especially with tools like CRISPR-Cas9, offers this incredible precision. Theoretically, you could correct specific mutations that cause age-related diseases, or maybe even tweak genes known to influence the aging process itself.
Sounds powerful. What are the roadblocks?
Several big ones. Off-target effects, accidentally editing the wrong part of the genome, are still a concern. Delivering the editing machinery to the right cells throughout the body, especially non-dividing cells, is tricky. There are ethical debates, particularly around germline editing. And aging isn't caused by one gene, it's polygenic, involving many genes, which makes targeting much harder.
Okay, complexity again. And telomere-targeted therapies. We talked about the telomerase dilemma.
Exactly. Telomeres shorten, that's linked to aging. Telomerase lengthens them, but too much activity risks cancer. So the goal is finding ways to perhaps transiently activate telomerase or deliver it carefully just to lengthen telomeres without letting it run wild.
A fine line to walk.
A very fine line. There have been some controversial reports, like Elizabeth Parrish claiming self-treatment, but those lack rigorous scientific validation. Some preclinical studies suggest telomere lengthening can have restorative effects, but balancing benefits against cancer risk remains the central challenge.
Seems like genetics offers precision tools, but wielding them safely and effectively for something as complex as aging is still a major work in progress. What about stem cell therapies, counteracting that stem cell exhaustion?
Yes, the idea here is replenishment or rejuvenation, trying to restore the pool of functional stem cells.
Hmm.
Mesenchymal stem cells, or MSCs, get a lot of attention because they seem to release factors that help with repair and calming inflammation.
Are these being used?
There are many clinical trials underway using various types of stem cells for specific diseases, things like osteoarthritis, heart failure, neurodegenerative conditions. The SENS approach also focuses on replacing lost cells.
But is that systemic anti-aging?
Mostly, no. The current focus is primarily on treating specific diseases or localized damage, not really reversing overall aging throughout the body. There are challenges with getting transplanted cells to survive and function properly in an aged environment, long-term safety concerns, ensuring they don't grow uncontrollably, and the type and source of stem cells matter a lot.
So promising for specific repairs, but maybe not the fountain of youth just yet. Okay, our sources also mention some really futuristic sounding things, nanomedicine and AI.
Yeah, these are really enabling technologies. Nanomedicine works at the molecular scale. Think tiny particles or devices.
Like tiny robots.
Potentially, someday. But more immediately, it offers ways to deliver drugs like senolytics or epigenetic modifiers or gene editing tools much more precisely. Target just the diseased cells, leave healthy ones alone, minimize side effects, maximize efficacy, or use nanoparticles for super early diagnosis.
Targeted delivery sounds key for safety and AI-driven drug discovery.
This is already making a huge impact. AI and machine learning algorithms can crunch massive data sets, genomics, proteomics, clinical data to find patterns humans might miss.
How does that help?
It helps identify new potential drug targets involved in aging. It can predict which molecules might work, even design new drug candidates from scratch, or find existing drugs that could be repurposed for aging.
Faster drug development.
Potentially much faster. We saw the example of in silico medicine using AI to rapidly develop an IPF drug. The open-air retro biosciences collaboration on reprogramming factors is another example. AI is also helping build better aging clocks and biomarkers to track interventions. BioAge is using AI on biobank data for this.
So AI is like a super-powered research assistant.
In a way, yes. Analyzing complexity, finding needles in haystacks, but it's not magic. It needs good data and smart biological questions.
Right. And lastly, there were various novel compounds mentioned, resveratrol, rapamycin, things like that.
Yes, a whole range of molecules are being studied. Sirtuin activators like resveratrol, flavonoids like quercetin and fisetin, mTOR inhibitors like rapamycin, even diabetes drugs like GLP-1 agonists. Many show promise in lab animals, often targeting those hallmarks we discussed.
But the human evidence.
It varies. Rapamycin definitely extends lifespan in many animal models, but it suppresses the immune system, which is a major drawback for widespread human use. For many others, the rigorous human data for actual anti-aging effects, not just disease treatment, is still limited or ongoing. Lots of research, but translating it is key.
Okay, so it's this incredibly active landscape, senolytics, reprogramming, genetics, stem cells, nanotech, AI, new compounds, a lot happening. Given all this, let's circle back to that 40-year question. Can science realistically pull off something like immortality in that time frame?
It's the billion-dollar question, isn't it?
Yeah.
And as we just discussed, AI and big data are definitely acting as catalysts. They're accelerating discovery, helping us make sense of the overwhelming complexity.
Right, the in silico example, the OpenAI collaboration.
Exactly. That synergy between AI, massive data sets, and automated lab work creates this positive feedback loop. It could significantly speed things up. But, and it's a big but, AI isn't a magic wand.
It needs the biology.
It absolutely needs solid biological understanding to guide it. Good data is crucial. Sometimes AI models can be black boxes. We don't fully understand why they make a prediction. David Sinclair made the point that bioinformatics is essential here.
Makes sense. Now, when you look at what the experts themselves are saying, there's quite a range of opinions on this 40-year possibility, isn't there?
Oh, definitely. A whole spectrum. On the very optimistic end, you've got figures like Aubrey de Grey famously predicting the first person to live to 1,000 might already be alive.
Wow, that's optimistic.
Very. And the SCNS Foundation talks about achieving engineered negligible senescence and reaching longevity escape velocity, where medicine advances fast enough each year to add more than a year to your remaining lifespans. Tech VCs investing heavily, like Altman and Retro, also signals strong belief in near-term potential.
Okay, so that's the high optimism camp. But then there are others who are more Cautious.
Much more cautious, yes. Many established aging researchers, like Luigi Ferrucci at the NIA, put the emphasis squarely on health span, extending the years of healthy life, not just maximum lifespan. We saw that Nature Aging study suggesting radical life extension this century is implausible.
They mentioned a slowdown in lifespan gains.
Yes, and a potential plateau in maximum human lifespan. Even researchers like David Sinclair, who are proponents of anti-aging research, have said we're still a long way from an immortal mouse, let alone humans. Some philosophers and biologists like Leon Kass argue there might be intrinsic biological limits, a kind of hard stop.
So real skepticism, too.
Absolutely. The World Bank suggested a physiological limit might be around 97, though they conceded we might manufacture lifespan beyond that. Andrew Steele focuses on health span and tackling that wall of mortality. And the general tone from major bodies like the National Institute on Aging is focused on health, well-being, preventing disease, more pragmatic goals.
Why such different views? Is it just interpreting the same data differently?
That's part of it. But it's also different philosophical viewpoints on what aging is, different levels of faith in technological acceleration versus biological limits, and different fields of expertise. A bioinformatician sees data patterns. A clinician sees the hurdles of human trials.
Which brings us right back to that translation issue. Getting from the lab bench to the bedside, it sounds like that's a major bottleneck, regardless of the specific technology.
It's huge, often called the valley of death in drug development for a reason. So many promising ideas fail at the clinical trial stage. It's long, it's incredibly expensive, high failure rate.
And are there unique hurdles for aging interventions?
Definitely. How do you run a clinical trial for aging? It takes decades to see a lifespan effect. So you need reliable surrogate endpoints biomarkers that predict long-term outcomes. Defining those is hard. Plus, the ethics of intervening in the normal aging process in healthy people. And the regulatory path currently, agencies like the FDA approve drugs for specific diseases. There isn't really a pathway yet to approve a drug simply for aging itself, so researchers often target age-related diseases as a proxy. All these things slow down progress considerably.
So even a lab breakthrough tomorrow, it could be years, decades maybe, before it's a real-world option. Okay, let's dig into some of those fundamental obstacles a bit more. This idea of a hard cap on human lifespan, is there a consensus?
Not really, no. It's still a major debate. The arguments for a hard cap often point to Jean Calment, the oldest documented person at 122. That record hasn't been broken in decades. Evolutionary theory suggests there wasn't strong pressure to select for genes promoting survival long past reproduction, and the Hayflick limit on cell divisions is seen as a potential built-in constraint.
Okay, arguments for a limit. What about against?
The main arguments against are the dramatic increases in average life expectancy over the last century or two, showing lifespan isn't totally fixed. And of course, those examples from nature, like hydras showing negligible senescence, is biologically possible, at least in some organisms.
So the jury's still out?
Pretty much. Our research suggests the current data just isn't conclusive either way, and that fundamental uncertainty obviously makes predicting something like immortality very difficult.
Right. Beyond that potential cap, what are the other big scientific or technical roadblocks we still face?
Well, safety and efficacy in actual humans, not just mice, is number one. We need robust proof that these interventions work and are safe long-term. Delivery is another huge one. How do you get these therapies, gene editors, senolytics, stem cells, to the right cells, all throughout the body, efficiently and safely?
Systemic delivery is tough.
Very. And the sheer complexity of aging, hitting multiple hallmarks, likely requires combination therapies. That adds layers of complexity in development, testing, regulation. Then there are the regulatory hurdles for targeting aging itself, which we mentioned. And finally, cost and accessibility. If these therapies are incredibly expensive, who gets them? That raises huge ethical questions about a longevity divide.
Yeah, a world where only the rich can afford to live significantly longer. That's a worrying prospect. All these challenges really highlight the scale of the undertaking. And throughout this, we've kept bumping into the distinction between health span and lifespan. Why is that so critical here?
It's absolutely critical, because while living forever grabs headlines, the immediate, pragmatic, and arguably more ethical focus for most mainstream research right now is extending health span.
The healthy years.
Exactly. The years lived free from debilitating age-related disease and frailty. The goal is to compress morbidity, shorten that period of sickness at the end of life. Let people live longer, yes, but live those extra years well. Active, engaged, healthy. That's what society largely wants and needs.
And helping health span might also increase lifespan a bit.
It likely would, indirectly. If you prevent or delay major killers like heart disease, cancer, dementia, people will naturally live longer on average. But it's a different goal than directly trying to break through fundamental lifespan limits, if they exist.
Okay. So pulling all this together then, let's try to answer that initial huge question. Based on everything we've looked at, the science, the challenges, the expert views, what's the likelihood? Can humans live forever or achieve radical life extension within the next 40 years?
Well, synthesizing all that, given the profound complexity of aging, the fact that many of these exciting interventions are still quite early stage with significant hurdles, especially in human translation, and the cautious perspectives from many seasoned experts, achieving true biological immortality by 2064 seems scientifically implausible.
Implausible for immortality. What about radical life extension, living centuries?
I'd say that also remains a very, very long shot within that 40-year time frame. The challenges are just too substantial right now.
Okay. So maybe immortality isn't on the cards for 2064. But does that mean the research is fruitless? What are the more likely and still valuable outcomes you see?
Oh, absolutely not fruitless. Far from it. The more probable, and I'd argue incredibly meaningful, advancements will likely be in extending health span and significantly improving how we prevent, manage, maybe even reverse aspects of major age-related diseases.
So better quality of life as we age.
Exactly. Imagine therapies that genuinely delay or mitigate cognitive decline, keep joints healthy, maintain muscle mass, boost immune function later in life, enhancing vitality, improving function, compressing that period of decline. These would be massive wins, huge achievements, even if we don't live to 500.
So maybe the story isn't immortality soon, but rather a radically healthier, more vital, older age is coming into view.
That feels much more realistic, yes. This 40-year mark isn't an end point. The research happening now is laying crucial groundwork, is building the foundations for whatever comes next, even if it takes longer.
And that idea of longevity escape velocity.
That remains a fascinating, but still theoretical, long-term goal.
Yeah.
Something for the more distant future, perhaps.
So wrapping up, immortality by 2064. Unlikely. But the pursuit itself, it seems poised to deliver potentially transformative improvements in how you experience aging, better health, more vitality, pushing back disease. That itself is revolutionary, isn't it?
Absolutely. Even without immortality, fundamentally changing the experience of growing older would reshape individual lives and society in profound ways. It really makes you think, what would you do with an extra 10, 20, maybe even 30 healthy, active years?